My dear students, in this class, let us learn about the structure and functions of chromosomes. Chromosomes are responsible for determining the physical and physiological and psychological traits of an individual organism. And these chromosomes are present inside the nucleus in case of eukaryotes, and in case of prokaryotes, they will be floating in the cytoplasm. These chromosomes are named so because they appear like highly colored bodies. So, like that, these chromosomes are responsible for determining the phenotype and also the genotype of an individual. Apart from that, they also determine the sex of an individual organism. Chromosomes are the vehicles of inheritance. Chromosomes means colored bodies. They are stained by fuelgen during interphase. Chromosomes were observed for the first time by Hofmeister. Hofmeister observed these chromosomes in the nuclei of pollen mother cells of Tradescantia. The term chromosome was coined by, of course, Waldeyer. So, chromosomes for the first time were observed by Hofmeister, whereas the term chromosome was coined by Waldeyer. In bacteria, because bacteria are prokaryotes by nature, in bacteria there is only a single chromosome. Number of chromosomes in plants and animals varies with the species. Total number of chromosomes present in the gametes is called haploid set. This is represented by N, and total number of chromosomes present in somatic cell of an organism is called a diploid set, and this is represented by 2N. For example, in case of human being, the diploid set of chromosome is 46, whereas haploid set of chromosomes in human being is 23. So, like that, in gametes haploid set is found, and in somatic cell, that means in body cell, diploid set is found. Haploid set of chromosome in any organism is called genome. Minimum number of chromosomes in animals is found in Ascaris megalocephala. That means Ascaris megalocephala, which is a nematode, it contains minimum number of chromosomes in the entire animal kingdom, and that number is 2. That means the number of chromosomes in Ascaris megalocephala is 2. And in animals, maximum number of chromosomes is found in radiolarians. Number of chromosomes in man is 46, in gorilla it is 48. Drosophila has 8 chromosomes, whereas a sugar cane has 80 chromosomes and a pea has 14 chromosomes. Normally, the plant chromosomes are bigger in size when compared to animal chromosomes. The chromosomes that determine the body characters are called autosomes. So, autosomes are nothing but body chromosomes. Allosomes are sex chromosomes. In human, there are 2 sex chromosomes, namely X and Y. Of course, chromosomes have the following parts, namely chromonema, centromere, secondary constriction, satellite bodies and telomeres. Chromonema are the thread-like or filamentous-like structures that are interwoven with each other in the chromosome. Centromere is the primary constriction. Secondary constriction is responsible for formation of nucleolus. Satellite body is found just beyond the secondary constriction, and the telomeres are the ends of the chromosomes. Based on the presence of centromere or based on the position and location of the centromere, chromosomes appear in different shapes and they are categorized based on the location and position of the centromere. Let us see all these parts. What is chromonemata, what is centromere, what is secondary constriction, satellite body, like that one by one we try to learn. Spirally coiled identical filaments in the chromosome are called chromonemata. Here you can see the spirally coiled thread-like filamentous structures that are interwoven very closely with each other, and these thread-like structures are called chromonemata. These chromonemata during the process of cell division they become condensed to become chromatids. So, there is no difference between chromonematia and chromatid. Chromatid is the highly condensed form of chromonematia. Chromosomes during interphase appear like filamentous coiled network, and this is called chromatin reticulum. Here in this diagram you can find this chromatin reticulum. And if you observe the complexity of the coiling of this chromonemata, if the coiling is easily separable like this, if the two coils are easily isolated like in this diagram, such coiling is called paranymic coiling. So, if two strings of chromonemata are isolated from each other very easily, then that coiling is called paranymic coiling. If the coiling is complex and if the coils or the strings are not separated easily, then it is called pectonymic coiling, that is called pectonymic coiling. So, like that there are two types of coiling of chromonemata, one is paranymic and another one is pectonymic. The condensed form of chromonemata during cell division is called chromatid. The shape of the chromosome is determined by the position of centromere. Centromere is the primary constriction, and this centromere is responsible for the attachment of chromosomes to the spindle fibers. Let me repeat, the shape of the chromosome is determined by the position of centromere. Centromere is the primary constriction and it serves some purposes. The centromere is responsible for the attachment of chromosomes to spindle fibers, and these centromeres also help in the process of generation of spindle fibers. Based on the location of the centromere, chromosomes are divided into four types. One is telocentric, telo means end. The centromere in telocentric chromosomes is located at one end of the chromosome. They are normally present in protozoans. So, telocentric chromosome is the chromosome where centromere is located at one extreme, at one end of the chromosome. They are normally present in protozoans. Telocentric chromosomes are very rare in animal kingdom because they are normally found only in case of protozoans. Here you can see at the end the centromere is located. Acrocentric chromosomes are found characteristically in locus, and these acrocentric chromosomes are rod like, and you can see the centromere not exactly at the end, little bit prior to the tail end of the chromosome. So, like that acrocentric chromosomes are rod like chromosomes that are normally found in locus. Submetacentric chromosomes are L shaped because of the presence of centromere in such a manner that the chromosome is divided into one short arm and another long arm. Long arm is called Q arm, whereas a short arm is called P arm. So, like that submetacentric chromosomes are the chromosomes which are L shaped. Metacentric chromosomes are the chromosomes that are V shaped, and these metacentric chromosomes are the characteristic chromosomes of amphibia, and they are V shaped. That means they are provided with equal arms. Previously in submetacentric chromosomes which are L shaped, we have seen two arms, namely P arm and Q arm, one short arm and one long arm, whereas in case of metacentric chromosome the arms are equal in size. Tip of the chromosomes are named as telomeres, and these telomeres are very crucial in the process of aging, and these telomeres are rich in G repeats. Enzyme present in telomere is telomerase. At each and every cell division the length of the telomere becomes short. That means the telomeres gets shortened with each cell division. Shortening of telomere causes aging. Even the lifespan of any organism is decided by the size of the telomere according to scientists, and this telomere contains one enzyme called telomerase. So, the length of the telomere decides the lifespan of any individual. And coming to other structures like secondary constriction. Secondary constriction is concerned with the formation of nucleolus. Secondary constriction is concerned with the formation of nucleolus. Hence, this is called nucleolar organizer. So, secondary constriction is known as nucleolar organizer because it gives rise to the nucleolus. Terminal portion of the chromosome behind the secondary constriction is called satellite, and the chromosomes that has satellite are called SAT chromosomes. SAT means sin acido thymonucleinico, that is the expansion of the SAT. The sequential arrangement of chromosomes of a cell is called karyotype. The diagrammatic representation of the chromosome of an organism is called ideogram. That is the difference between karyotype and ideogram. Karyotype is the sequential arrangement of chromosomes depending on the size, that is called karyotype, whereas the diagrammatic representation of the chromosome of an organism is called ideogram. So, that is about the structure and various complexities of the chromosomes. And coming to the functions, chromosomes are the vehicles of inheritance. They decide the phenotype and also genotype. They also decide the sex of an individual. Formation of new species is triggered by the mutations that occur at chromosome level, and formation of nucleolus, because I told that there is one constriction called secondary constriction within the chromosome that is responsible for the formation of nucleolus, and for all metabolic activities chromosomes are needed. So, these are the functions of chromosome. So, let me sum up. Chromosomes are vehicles of inheritance or vehicles of heredity. Chromosomes were first observed by Hofmeister, and the term chromosome was coined by Waldeyer because of the colored nature, and they are being stained by G.L.N during the process of cell division. And chromosomes contain certain structures called chromonemata, centromere, telomere, secondary constriction, like that. Chromosomes decide phenotype and genotype of an individual, and centromere position is very crucial in the classification of chromosomes, and secondary constriction is responsible for the generation of nucleolus. So, thank you very much.